Good afternoon, everyone. My name is Abhishek Narain Singh. I'll be presenting a talk titled Customized Biomedical Informatics, or the role of computer science and logic and informatics that is used for biomedical informatics. So the previous speaker gave a talk about the assembly and how to track information. So if we take on those lines, we have, as we know, putting everyone at the same level, we have DNA as a blueprint of life. It's comprised of A, T, G, and C. And when we combine all the possible set of A, T, G, C in a cell, whether it's from proteome or whether it's decided on organs such as mitochondria or from any other part of the cell, the whole set combined together, assembled, is called the genome. And whenever two people are compared, two people will have differences in their genome. So this is not a good way that we start comparing only two individuals. The more practical solution is that we keep a benchmark genome as a reference. And whenever someone is to be compared, his genome will be compared to that benchmark reference genome. Currently, we are using the NCBI build 37 point something as a reference genome. And that reference genome also keeps on updating, so we have to always keep the benchmark in our mind when we compare it. So typically, if we have a sequence of reference genome, then if we align this individual genome, there is a change in the basis in all references. If it was present earlier, then it can be polymorphic. If those bases from the reference genome are removed compared to the individual, we call it deletion. And if those bases are introduced, then we call it an insertion. And there can be actually multiple ways by which these things can happen. I'll say this again. So in the genome analysis, what we have is a sequencer machine. It can sequence a DNA or even an RNA, but RNA is supposed to convert it to cDNA, complementary DNA, to then sequence it. And what we get is list of small reads or sequences, and that can be then aligned to a reference genome. So these are the reads. These reads can be typically as small as 30 bases and can be high as 300 bases, or with the older technologies it can be maybe in 1,000 bases long. Because the technology does not exist to sequence the whole genome at one go, because of the chemistry involved and the quality of the bases that appear eventually. So we have to depend upon the existing technology, and those the reads are very small, and since the cost of sequencing is going down exponentially, what the strategy adopted is that these reads are multiple sequences. So for any given region of the genome, the sequences will have multiple copies. So let's take it further. I took out the first region and put it over here and found an exact match. I took out the second region and put it over here. There were substitutions at certain points. Like DNA got substitution, GMC got substitution. Took out the third region and set it over here, aligned it over here, and I got an insertion at certain region. And then similarly, there was 3 removed when I compared the next sequence. So essentially, when we align the reference, the genome sequence of an individual to a reference sequence, all kinds of variations can happen, which are broadly classified as structural variations and SNPs. SNP stands for single nucleotide polymorphism. That's a point mutation point that we talked about. And structural variations can be further classified as insertions and deletions, and inversions, copy number variation. And these insertions can be novel, that it is not present in the reference genome, and it can further classify into truly novel, which is not present anywhere in any organism database, or just organism-based novel for that individual. And there can be further classification. Let's not get into detail at the moment. So there are a lot of approaches. One of the approaches which the previous speaker also talked about is the assembly approach. There are other approaches, the reads that we get. There can be front end, there can be a back end for a region that is sequenced. The sequence quality reduces as it progresses along the length. So we need to compromise at what point we want to stop the sequencing to take place. So typically, like Illumina company sequences, say 101 bases at both ends. And it can actually go beyond, but it's not going to be useful because all of the information that we get might not be reliable. So what we essentially adopted is we have a chunk of the sequence of the DNA, we read the front end, we read the back end, and we have an approximate idea of what is the insert length in between. So based on this information, we can align them to representative sequence centers, and every alignment happens in a fashion that we would not be expecting. Then it can be a deletion or an insertion as the case be. Similarly, we know that the genome is sequenced 12 times on average on every region, and we find that a certain region aligned on that is having more than 12, maybe 24, then we know that this is a region of duplication or maybe copy number variation. And there's still a split read method where a read itself is split at certain region being aligned to another region and the other region to another region. And there are many of those. It is of the coverage. And this is called delineation. So based on that, a lot of the softwares are available openly, and I also developed certain softwares. One is called the MSET, Massive String or Sequence Exhaustive Comparison Tool, which has been finding out the copy number variations, and this is the MSET plot. Different colorations during the percentage identity that we got, and there are lines in between which take the not present in one genome and present in another genome, these kind of things. And the genome break, which doesn't look much into the copy number variation, just looks at the structural variation of insertion, deletion, and steps. So this plot is essentially some combination of insect and genome break plot. And like the previous speaker, I also compared the cellular sequence, which is the JPEG Venture's genome 50%, 40% coming from other individuals, to the NCBI 36 genome assembly at that point. And until a decade ago, in 2003, we thought that the genome of any two random individuals would be 99.9% similar. So when I did the analysis of cellular versus the 36, I realized that this percentage doesn't seem to be what I read in the textbook or what other people were saying. It doesn't look to be correct. And so I reported people I worked for, and they published a paper that the genome is less than 99.9%, approximately 99.5% similar of two random individuals. It was a bit upsetting because my name was not there in the paper in 2007. So I did a further analysis and published another article that the genomes are less than 99.5% similar based on the comparison of cellular analysis. So essentially, when these analyses are done of number variation of certain loci or region of interest.
So finally, what is the approximate number which is common?
We can put the question later on. So the loci of our region can be then mapped, which is what is represented in a specific fashion. We can know about getting different information. So, variation. Why everyone is talking about what the genome mapping, proteome mapping, all these things, bioinformatics, computer science. Why is it so can get so hot topic? Why are we sitting here? For simple reason that discovery, we know that these certain genome markers are associated with certain genes. We can make certain analogy with another people's genome analysis. We've also heard about GWAS, genome-wide association studies. So those kind of things can be done. Ancestry tracing. If I have a variation, probably I got it from my father or mother, or whatever the case be, and they also got it probably from their father and mother, so we can trace it in certain fashion. So we can, the variations can be harmless if it doesn't affect the regulation of the gene or the coding section of the gene itself. Or it can be harmful, such as those which are fairly established, such as BRCA1 and BRCA2 in the breast cancer, And then there are heart diseases, diabetes, Huntington's diseases, a series of autism, a series of schizophrenia. All these kind of diseases are really very acute diseases, and there has been a lot of genetic research done trying to link the markers that can be attributed to these diseases. And these can be actually latest as well, that they can become active only under exposure to certain physical external conditions or on the intake of certain kind of food habits and living habits. People can be carriers that they are not actually diseased persons, but when they mate with someone else, obviously opposite sex, and the offspring might combine to get the disease. So those kind of information can be extracted from this kind of genome analysis. And of course, they can also be recently, people have also started to look at the traits and characteristics extraction. Why people at all? Why people? Some people can be obese. So essentially, to put it in the form of one picture, we have a medical doctor who is also a bioinformatician or biomedical scientist. He can categorize people into subgroups like this and advise them accordingly, or give them a personalized, customized medicine or treatment or suggestion accordingly. So we are all living with our near and dear ones who are suffering from one of the major diseases. Some of the common diseases are like autism, diabetes, cancer, schizophrenia. And there are so many people around us who are suffering from chronic diseases. And even the people who are called healthy, they might also sometimes get some diseases or the other. But there is definitely a misuse of this variation that can be done. And such as a health insurance company, if they get to know that this person is likely to get affected by certain diseases, they will jeopardize this insurance. The employment, human resources, they will also keep track of this. These are things we have to be aware of. And relations like marriage, or people, you get to know about your girlfriend or boyfriend, if he's going to have certain kind of genetic problem eventually, that might also be an issue which people take informed decisions. There's also a concern that people who are not that educated, they might misinterpret the genetic results, variation results, and which a disease which can actually happen at some stage, or he might interpret that, I'm suffering from this disease, and it can be a psychological issue. So these are the things that we need to take care of. So typically, this genome analysis is the only solution? No, it's not. Because if we have an identical twin, someone is raised in America, another person is raised in China or maybe India or Europe, it's not just the genome, it's your upbringing, how your neural network got tuned in, what your background is, what the exposure is. So it's not a single silver bullet that solves everything, but it does give us clues for certain directions. So genome brain was used for comparing a family of 4 people: the father, this is A105A, the mother, B, C is the daughter, and D is also the daughter. And it was compared to a reference genome, and the result of the genome-based software. And using this, we can extract out the SNPs and variations. Zoom in a bit. If I zoom in, these are the SNPs associated with certain loci, index at certain loci. And these are the SNPs at certain loci of interest. And if we do a population-wide analysis, such as this, we also found out a northern region in the Dutch population, which is not present in any database. So we can link this particular northern region to Dutch-specific population. And that was this informatically, bioinformatically, and it was confirmed by better PCR techniques. And for example, I simply take out a fetal DNA from another group and compare the Y chromosome SNPs. So we find that the SNPs found in the father is far higher than in the mother, and in the true child, the SNPs are compared to that found in the mother. So we can actually say that the child which is to be born is going to be female. So You don't need to do expensive and, maybe injurious, sex discrimination technology, otherwise it can be a waste. So instead of SNP, this time I took this FV insertion deletion, and I found that the variations are comparatively much higher. The differences are much higher. So from this we can propose that the structural variations are more restricted. They have more selection pressure to changes, and structural variations such as FD or insertion and deletion, they are more characteristic in nature for trade, health, whatever than the SNP. So here is an example. I took the mitochondrial DNA of the four family people: father, mother, and the two daughters. And it is known in literature earlier that the mitochondrial DNA is maternally inherited in humans. There has been a case reported that mitochondrial DNA can be even paternally inherited, such as in banana, but never in humans. But I'm proposing that it can be possible that mitochondrial DNA can be even paternally inherited, because here is a SNP found in father and is found in one of the mother, one of the daughters, and also the mother. So this is the power of informatics, which people have to do extensive, experimental design of microscopy and all those things, and then they came up with the conclusion that the sperm hybridizes between the ovary, and the tail goes off, and that mechanism sometimes fails, and so mitochondrial DNA can be sometimes inherited even from the father. So this was the discovery I made for the first time for mitochondrial DNA. And there was also one structural variation found in the mother and one daughter, and not found in one of the daughters. That was also very disturbing to know. So in conclusion, we get to know that the genome file fingerprint can be a more deterministic and more cost-effective, and at least it can guide us to a certain direction when we are doing the analysis. It can, such as the sex discrimination, and I also propose that mitochondrial DNA can have paternal leakage of inheritance. And one thing I didn't talk over here, but I can tell you now, that whenever we do organ transplants, like converting kidney transplant or something like that, there is a fair possibility of that organ transplant to go as a failure. And the reason for that is the immunological effects, consequences that result from it. So that means that if we can do a pre-analysis by looking at the MHC domain, which is responsible for immunological effects in human beings, that is HLA, loci, or chromosome 6, and we see that these two people don't have much of the variations in the MHC region, then that's a good hint that this organ transplant can be successful. So that's the power of analysis, where we are all getting towards more artificial intelligence in these things. This thing I told repeatedly, that variations can be mapped to traits, phenotypes, behavior, etc. with a degree of confidence. We can't say that the systems are good. If you have this test, you are going to suffer with this. There is certain confidence based on what we already have observed from the database that such a percentage of people have been reported for this kind of phenotype. And the last thing that I mentioned, that structural variations are more specific characteristics of an individual. They are more a characteristic signature for a trait or disease that we take than the thing. Okay. That is the conclusion. These are references from my own publications and some from the other publications, which is not from the I mentioned. And that's my contact email ID if you want to get that to me. And that's my website, where you get to know more about the software and the work that I do.
Anything? It will be next.
What percentage of genes, according to you, are common among various individuals?
So, the percentages that have been reported, those are to random individuals. Obviously, when you take a very unhealthy person who is suffering from lots of diseases, for example, say hypothetical person who is suffering from liver disease, heart disease, diabetes, any number of diseases as one suffering. Okay. then obviously you will have big sections of genome or gene region, gene regulatory region that will be missing. And if I compare that individual with a healthy individual such as you or me or anyone else, it might be like just 80% or 80%, and it might be even 95%. But two random individuals we talk about, healthy people, we get an estimate that, okay, if it was 99.5%, I say it can be roughly less than 99.5%. More questions?
Engage with this research yourself.
Everything that I reported has been done by me. Something that I've been engaged in genome research off and on since like 2006 onwards. Yeah. So it's like compared to some work done when I was in Canada, and I was also working in the genome genetic project. So it's like a combination of my friends.
Data are available for various diseases.
Yes, and that's there's such as a DP set, which is an openly available thing. But some people like to keep it private data. But private hospitals, they don't want to release to the public.
Okay, thank you.